Welcome to Challenging Climate, a podcast where we discuss the science, technology, and politics of climate change and more. I'm Jesse Reynolds, an environmental policy expert.
I'm Pete Irvin, a climate scientist. Each episode we bring on a guest with a unique perspective and deep expertise on climate change, or something else, and put challenging questions to them.
In this episode, we spoke with Ben Novak, who is lead scientist at Revive and Restore, which, as you may have noticed from our introduction to this episode, most of its work falls out of climate change directly because there he leads the de-extinction efforts. And the overall theme of our conversation was the use of biotechnology for conservation purposes. And this covers a number of proposed methods and proposed objectives, not just de-extinction, but also genetic modification of wild species and assisted evolution or assisted adaptation, in which biotechnology or traditional breeding is used to help wild populations of species adapt to a changing climate.
Now, Jesse did this episode alone, and so he carefully avoided the reference that I would have jumped to immediately, which is Jurassic Park, de-extincting dinosaurs, and the fact that, well, that's sci-fi. But here we're talking about the nearly almost reality of being able to bring species back from the brink, more recent species. If you go too far back in time, I think the DNA is too damaged to make this practical. I think in a way, this de-extinction effort is will be the crowning achievement of biotech for biodiversity purposes. But they discuss a much wider range of things, from fertility treatments developed in humans being applied in breeding programs, and a whole range of ways that do connect up with climate change, of efforts to enhance the adaptation potential of species that are struggling to deal with climate change. I thought it was a great conversation. And yeah, without such radical interventions, such radical transformations in the way we interact with nature, it's going to take quite a beating with climate change. So it's an interesting side issue, but related to climate change. I thought it was a great discussion.
And another way that we bring it to climate change, and specifically to the research interests of Pete and myself, is that Ben Novak, our guest, helped lead a workshop and subsequent brief paper that was published on the idea of intended consequences, an effort to look at intentional interventions in natural systems, in this case, biological systems that biotechnology could be used to intervene in. And it's a call for an appropriate balance of thinking about not just unintended consequences, which of course are often the focus of popular and academic speculation, but also not to forget the intended consequences of these activities. And Pete and I, of course, have worked in the areas of, to a lesser degree, carbon dioxide removal, but especially in solar geoengineering. And these questions of intended and unintended consequences loom large in both fields.
Yeah, I think this issue of, if there's a possibility, an option that's on the table or could be put on the table that could potentially avoid something really bad happening, is it better to avoid that because you're doing something novel, something frightening, and have that terrible consequence happen, or to step in and do something about it in a way that's novel, perhaps a little frightening, challenging, but which is the only reason you're doing it is that the threat of the world going on as it is big enough. So yeah, I think there's a nice parallel there with geoengineering.
And now, Ben Novak. Today, we are joined by Ben Novak, who is lead scientist at Revive and Restore, where he is its de-extinction leader, especially focusing on the group's restoration of the extinct passenger pigeon. He is also the lead coordinator for its conservation cloning projects and the program manager for its new Biotechnology for Bird Conservation endeavor. His work at Revive and Restore includes extensive education and outreach, co-convening of workshops, and developing projects and scoping opportunities to expand genetic rescue science and applications. Ben, welcome to Challenging Climate.
Thank you for having me. I'm really excited to be here.
So we like to begin by helping readers understand who you are. How did you get to hold what strikes me as something of a peculiar position? What's your background? What led you from, high school or college to working on de-extinction?
Yeah, my completely made up job. Yeah, so I am from Western North Dakota. born and raised near Theodore Roosevelt National Park. And for anyone not from the Dakotas are similar to Wyoming and Montana and sparsely populated, rugged terrain. On the Great Plains side of things, it's agriculture wherever it can be done. But the unique thing about growing up where I was at in North Dakota, being near the park, was that Theodore Roosevelt National Park, while it's not the first national park, it is the place on earth where Theodore Roosevelt once lived, had a ranch, and was truly transformed into the conservationist he became. It's there where you see in his letters and his diaries that his viewpoints on preserving land for its aesthetic and species for their value unrelated to people truly starts to come through, which is, it's an awakening many people had at the time. Whereas he actually went out to North Dakota hoping to hunt bison and have that great kind of American safari experience. And when he got there, there were no bison to be found. And so then, he went about trying to make a living on that rugged landscape, and it was not very easy, but it's a beautiful place. It's one of the most beautiful places in the world, particularly if you're fond of deserts and things like that. It was stunning for me to be there, and living there not only exposed me to that kind of history of human mentalities changing from let's take advantage of nature to let's preserve it and help it, but also the concept that you can't just set aside land and say, okay, this is protected now, but you have to do a little more work than that. And if you go to Theodore Roosevelt National Park, you will undoubtedly see bison. If you're lucky, you may see an elk. And if you're very, very lucky, you go there many times and spend some time, you will see a bighorn sheep. Each one of them had gone extinct in that region in the early 1900s. And so Theodore Roosevelt National Park was set aside and protected, but to actually make it the ecosystem it is today, that it once was, conservationists brought bison in, they brought elk back in, they brought bighorn sheep back in. There was intervention and work and labor that went into trying to recreate that ecosystem. And so from a very young age, I was immersed in that. I was a lover of science, and around my really formative years in junior high, that's when the human genome was being sequenced. That's when the connection between birds and dinosaurs was on fire because of the feathered dinosaurs being discovered in China. That was when some scientists in Australia took a piece of thylacine DNA and put it into a mouse and showed that it could work. And there were articles about whether or not de-extinction could be real someday. It was also a time when a scientist for the first time genetically engineered mice. He knocked out the myostatin gene and they became more muscular and longer lived. And the Olympics was dealing with, oh my gosh, we need to ban gene doping before people can engineer themselves. There was a lot of buzz in the news about genetics and where it could go. And I was very, very fond of the extinct species, the paleontology news, the thylacine news. I'd grown up wanting to be a paleontologist, even digging up fossils in those rugged badlands of North Dakota. And then all of a sudden, here are these people getting DNA from an extinct species and people talking about whether or not we could recreate them. And knowing that in Theodore Roosevelt Park, people had restored bison after decades of being gone, had restored elk and bighorn sheep. I was not very far away from Yellowstone Park, where wolves had been reintroduced just several years earlier, and that was still, in big news. The concept that conservation works to restore species and ecosystems, to me, was cemented in my mind, and the prospect that biotechnology could take us to species that are completely extinct is what really fueled me. And so by age 13, I said to myself, I was going to use biotechnology to bring back extinct species. And the first species I turned my attention to was the dodo bird. I did a science fair project on whether or not we could bring back the dodo bird, sent me to state science fair, won my category division, and got a year's supply of Scientific American, which is what every kid needs to become interested in science, as well as National Geographic. And in that work, I learned that the dodo bird happens to be a pigeon. That was at the exact time in like history that Beth Shapiro had sequenced the first dodo bird DNA and compared it to other birds and found out, no, this isn't some relative of pigeons. The dodo bird is actually a pigeon. And so, that got me really going. I wasn't a pigeon obsessive aficionado at the time, but my grandfather raised pigeons, and I just discovered that dodo was a pigeon after this whole project on whether we could bring it back. And I started delving into more books about recently extinct species, and lo and behold, fell in love with a photograph and the story of the passenger pigeon. And years later, after being a graduate school dropout more than once, was in Canada working on actual passenger pigeon DNA with Hendrik Poinart at McMaster University. And that was my side project at the time. I was really trying to get it going. And for mental health reasons, struggles with depression and many things that I've had my entire life, I was not in a good place. I was ready to stop graduate school. And coincidentally, at that very time when my life was hitting its lowest, Stuart Brand and Ryan Phelan formed Revive and Restore around the crazy idea that biotechnology was finally at a place where we could recreate extinct species. And Stuart chose the passenger pigeon as their first project. And because I was tied into all of the other people obsessed with passenger pigeons, the 7 of us, I got in touch with people that attended Revive and Restore's first meeting. And Ryan and Stuart sent me an email the next day, and they were like, would you like to work with us? Because I sent them an email. I was just basically like, I have sequenced passenger pigeon DNA. You can have it. We had a 1-hour phone call, and 10 years later, I am in this full-time position, having helped Ryan and Stuart build Revive and Restore from a passenger pigeon project, a moonshot for biotech, to what we had originally envisioned as this spectrum of a genetic rescue toolkit of everything from helping species through genomics and the information it brings to reproductive technologies to the prospects of gene editing for species. And yeah, today we now support over 40 projects in 15 nations. I personally lead passenger pigeon and heath hen de-extinction. And as you said, I'm coordinator for our black-footed ferret work, liaison for the Przewalski's horse work. Those are cloning projects. And I am currently working to onboard at least 8 projects, our first projects for the Biotech for Bird Conservation program.
So here you are, you got your, sounds like you have your dream job after some ups and downs. That's encouraging. So we're going to talk today about the scope of work that I think of as biotechnology for conservation purposes. I don't know if there's a better phrase, but I'm trying to capture 2 primary techniques we'll talk about today. A little bit about de-extinction and the prospect of genetically modifying wild species for conservation purposes. So let's start with de-extinction. What is it exactly? How might it work? My understanding is there's a range of techniques, and the vision is to combine these and approach de-extinction. Am I on the right track?
Well, yeah. So of course we'll start with the most ambitious. On paper, de-extinction is really simple to spell out in terms of steps, and in reality, there's many nuances, and lots of people have published on what de-extinction is. It's just to clear up right away as a term, it's sometimes confuses people because they think we're resurrecting extinct species. When really de-extinction is more literally undoing the impact of extinction. And at Revive and Restore, we're ecosystem focused. So in the past, as I said, when you wanted to restore a locally extinct species like bison in an area, you went to where bison were still surviving, you took a few, you brought them back. People started hitting roadblocks. They on certain Pacific islands, Indo-Pacific islands, where they had giant tortoises that were driven to extinction. It's an isolated island. There was nowhere, they were completely extinct, nowhere to get a living member of their species. But people started noticing, there's a lot of convergent evolution between some of these giant tortoises. So they would borrow a giant tortoise from another island that might be millions of years diverged, but was taking up the exact same ecological role. And they've done that a few times, taking an unrelated species of tortoise, bringing it into an island, and that's called ecological replacement. So, but of course, both of those things are going to be pretty limited. If you're trying to restore the role of a species that was completely and utterly unique, where there's really nothing alive today that'll fit that role, what do you do? And this is where de-extinction in biotech comes in. So the concept is that there's 3 technologies that have come together today to make this possible. Something we call paleogenomics. People refer to it a lot as ancient DNA. So now we can actually take a piece of tissue from a stuffed bird in a museum, or a bone from permafrost in Siberia and get an entire genome's worth of DNA in it. It's fragmented, it's difficult to work with. There's a lot of limitations to how we can actually make sense of it, but ultimately we can get a really nice picture of the genetic code of an extinct species when we do the work the right way. We compare that code to its living relative, closest living relatives. Oftentimes you got to figure that part out first. So you use the DNA to even figure that out. Get the whole genome of a living relative. In the passenger pigeon's case, it's the West Coast band-tailed pigeon here in the USA. And you start identifying all the little parts where they're different. And in 2012, the technology that really made this possible was gene editing. So people had first sequenced the first extinct genome in 2010. And then 2 years later, the technology came around that, once you find these differences, you can now design CRISPR-Cas9 and its various now derivations to target those sequences in the living animal and change them to the sequence of the extinct one. It's very different from what genetic engineering has been for decades, whereas you just make a cut and splice something new in. This is fundamentally rewriting the code. And so the idea is, in a petri dish, you take some cells from that living animal, you make all these edited bits in the DNA, you now have a genome that's very close to your extinct species, and now you have one third last technological field to employ, and that's the reproductive technologies. When it comes to our birds, we can't use cloning because of the way they lay eggs, but there's over a dozen different types of reproductive technologies that have been developed with biotech, all to try and confront the different types of ways that different species reproduce. And so if you can figure out the reproductive biology in a good way to turn a cell into an embryo, into a whole organism, then you're finally in the terrain of you've given birth to this new recreated extinct species. It's new, it's a hybrid, it's old, it's a lot of philosophical things all at once. But then you start breeding them in captivity and returning them to the wild the same way that people have done for dozens of species that have been saved through captive breeding and conditioning and get them into the wild. And those are the steps we go through. And it really opens up the door for de-extinction to just about any species we can get that old DNA from, which are things from up to about maybe 100,000 or so years ago, depending on how well the specimens are preserved. And then the caveat are, do they have a close relative we can work with today? And do we have a reproductive technology that can work for that? And that's coming along, right? So like a good way to think about it is we could actually probably recreate a giant ground sloth, but living ground sloths are too small to give birth to giant ground sloths. So until someone creates a truly synthetic uterus, that's a species we can't do. There are other species that we could possibly get DNA from, but they're so bizarre and unrelated to anything today that we might not know actually how to recreate that sequence, what close living relative would work. And so, there's a lot of limitations, but for a species like the passenger pigeon, we've got this great close living relative. There's a lot of biology about how to raise pigeons, how to restore them in the wild. So the one real linchpin is really advancing the reproductive technology, which is kind of where I'm at with things.
Could you pick a species where work is being done? It could be the passenger pigeon or the black-footed ferret or whatnot. Could you give us a sense of where the research is at? How close are you and your team to successful, what you would consider to be successful de-extinction?
Well, we're a ways away. It's not happening next week or next month. I won't be having any surprise press releases. But we've been working on this project for about 10 years. And really the real limiting factor is not the science. It has been getting sustainable funding. We're a philanthropic group, a nonprofit. And so it's actually the idea of biotechnology for all conservation has resonated greatly with many funders, but de-extinction has not resonated with many. And so, this is so brand new, the concept, that a lot of people don't see the connections to regular conservation. So we've had a lot of campaigning to really do. And, really luckily, in just the last year, I think the world's readiness for these things is starting to change. So we did have some money come in from an anonymous donor, not simply to make passenger pigeons, but really to just overcome these challenges related to these bigger conservation goals. And it's what's enabled us to do the bird program now. And so where we're at for the passenger pigeon, we've sequenced their genome. We've sequenced the band-tailed pigeon's genome. I've been doing refined research on their natural history. We've studied the husbandry of band-tailed pigeons. We've obtained a captive flock of band-tailed pigeons for our purposes and some volunteers to raise them. So we've got a lot of resources and a lot of knowledge for just about all the steps, the breeding and things we need to do. I've personally developed the egg handling and manipulation techniques to be able to create an edited pigeon. But what we really need are the complete kind of efficient reproductive biology. So our Biotech for Bird Conservation program is aimed at solving these reproductive issues for birds, taking technologies that work really well with domestic chicken and finally get them translated to wild species, and possibly innovate completely new technologies that will be more compatible with the resource constraints and the needs of conservation of wild species. This is going to open up the door for a lot of things. As you said, not just de-extinction, but possibly gene editing wild species for conservation. One great example of that would be the Hawaiian honeycreepers. So Hawaiian honeycreepers are a group of unique, amazing birds. Over half of them are already extinct due to the effects of colonialism on Hawaii. And now the remaining species are drastically doomed by climate change because a couple hundred years ago, Europeans brought mosquitoes to the islands. And those mosquitoes were not necessarily a big threat themselves, but Europeans also brought with them some poultry and some other birds. And those birds carried avian malaria, and the mosquitoes then transmitted that malaria to wild birds. And the wild honey creepers of Hawaii, while some of them are now showing a little signs of resistance, the majority of those species had no predisposed resistance whatsoever, no lucky mutations, and have been annihilated by that disease, as well as a few other mosquito-borne diseases like avian pox. And literally, I heard the estimate that there's at least 4 species right now that are most likely to go extinct in the wild in the next 4 years. And if they can't get successful breeding programs in place, then they will be completely extinct due to climate change. Because as the years get warmer and warmer, the mosquitoes are able to breed for a longer season and go higher and higher in elevation up the mountains of Hawaii. So there will be no safe refuge. Now, biotech offers 2 solutions here that no other thing would do. One is effective ways of getting rid of the mosquitoes, which should be and is a priority. But the second is possibly gene editing the birds to carry the resistance alleles that would allow them to survive in a world with malaria. The reproductive technologies and gene editing technologies we are going to be advancing in our program are what will make a future like that possible for the Hawaiian honeycreepers. And that, of course, is the exact same kind of technological basis that will allow us to also recreate something like a passenger pigeon. So the 2 things go hand in hand. In conservation, the gene editing endangered species to cope with change is called facilitated adaptation.
So I'm a little curious, how is the genetic editing of a wild population done? Not so much what I mean in the lab, but in terms of the wild population itself. So you grab a few individuals from the wild population, genetically modify them, and do you put them back into the wild population? Would the genetically modified trait necessarily catch on, or might that trait then dissipate during selection, or the proper term is just drift?
Yeah. So from the population dynamic standpoint, the real idea here would be is you get a few of those organisms breeding in captivity, so that you can do the gene editing. And then rather than release the first few gene edited animals back out, you would then breed them up to get a larger number of animals possessing the gene edit. Now, if it's something like disease resistance, when you release those animals to the wild, there should be heavy selection for the new trait. So drift should not work against it. There are some people that have postulated that we should be adding gene drives to these things just in case, so that drift doesn't get rid of the gene. I actually don't feel that gene drives, to clarify, a gene drive is an element you add into your gene where it makes it so that it ensures that that gene is inherited in offspring. It breaks Mendelian inheritance and it tries to drive something from being inherited with a 50-50 chance to getting 100% inherited, and then it duplicates itself in the genome so that the offspring also spread that. It's a wonderful approach to thinking through ways to eradicate the mosquitoes and possibly control other invasive pests, as you could introduce a deleterious gene which would normally be selected against, but this would fix it and spread it and break that selection, and then you could get rid of that species. However, with a species where you're trying to introduce a beneficial trait, I personally don't believe gene drives are the right way to go, simply because the point of introducing beneficial traits is to actually allow selection to work. And while you don't want your species to go through a bad genetic bottleneck, that puts the onerous work on the conservation side of getting enough individuals from the wild population, going out and sampling, knowing what the genetic diversity is, who the lineages are, getting a couple animals from each genetic diversity pocket, getting those into captivity, getting those edited, and then introducing the full spectrum of genetic diversity, the effective population size back, and getting enough individuals back. Basically, you would maintain your lines of edited animals in captivity so that you can monitor the population in the wild. And as you keep supplementing these gene edited animals, you start measuring next generation. Is this trait going up, going down, staying the same? Do we need to supplement more males, more females? What do we need to do in order to finally get that moving towards fixation? For extremely endangered species that are rare, this may not be so difficult because there's not high numbers to deal with. But, luckily, the concept of gene editing entire species to save them probably isn't going to be something that we need for a lot of species. It is probably something that's going to be needed for at least one species of almost every ecosystem. That's the situation we're in the world right now, particularly with climate change and the spread of invasive species and disease. But when you target the few species that are most vulnerable and get those being very resilient, odds are we don't have to do this a lot. So that's the hope. But yeah, it's going to be quite laborious.
So would it be accurate to sort of summarize what you were describing and say, well, if the trait you want to introduce is highly advantageous to the population, such as resistance against a very deadly disease, not much of an intervention is needed. A small percentage of the wild population could be the genetically modified species. And as you move towards, let's say, a more neutral trait, perhaps resistance against a relatively uncommon disease, then a more forceful intervention, such as a higher percentage of the population, would need to be genetically modified. And then as you start to move over to the idea that you're actually trying to harm the species, reduce or if not eliminate populations, you need even more of a kick. And that's where gene drive can come in.
Yeah, and the beauty of gene drive is if we were graphing this, we'd actually see the inverse relationship, right? Like introducing a few individuals because there will be strong selection, then needing to introduce many individuals because there will be possibly neutral selection. When you get to the gene drive effect, you may only have to introduce then a few animals again because it's breaking the selection curve in its favor. And that's what's very attractive with invasive species, because of course, if your invasive population is truly damaging, you don't really want to have to release a whole bunch more to try and get to zero, because more of that species is bad on the environment.
So this podcast that you've joined is typically about climate change, and we haven't spoken much about it in this episode. You've mentioned it a couple of times in the context, for example, of the increase in avian malaria. Are there examples where assisted adaptation through genetic modification appears to have potential to help a species, if not an ecosystem, withstand climate change?
Well, right now it's only theoretical. At the moment, there is only one wild species that has been genetically modified in any way for a conservation purpose, and that's the American chestnut tree. The American chestnut tree was once an incredibly abundant tree in the eastern Appalachian chain here in the US, and it got hit by a disease introduced from Asia on ornamental chestnut trees, as humans have just moved, things around like mad in the world and brought diseases with them, and just got wiped out in just the weirdest way. So this fungal disease, a fungal blight, kills mature adult trunks of chestnut trees, but it doesn't kill the root system. So there are about 400 million root systems of American chestnuts that are still around. And every year they shoot up new trunks, little shoots, and those make it about 3 to 5 years, up to about maybe 10, 15 feet high. Skinny little polled trees, lots of shoots. And then the fungus just kills them back down to the ground. And so it's a species that's constantly dying. which is a really weird purgatory to be in. A team of scientists found a gene after discovering what it is that allows the fungus to live and kill the tree. The fungus is getting into the bark and excreting an acid, and that's how it's getting in there and causing its damage. A lot of different types of plants actually have enzymes that break down these acids, but the chestnut's gene for that job had broken a long time ago because it wasn't necessary in its life. So they grabbed a gene from wheat, oxalic oxidase, put it into the chestnut tree with old school genetic engineering tools, and created a line of chestnut trees that are completely immune to this disease. And in a way that works beautifully with the intended consequence of getting it into nature, because it's not killing the fungus, it's actually allowing the tree to coexist and live with the fungus. So there will be no pressure on the fungus to evolve, to have more virulence and become harsher. The fungus can keep living, so can the tree. That engineered tree is currently going through government regulatory approval to be basically released to the wild. It's going through deregulation so that people can buy seeds, plant them in their yards. We can start putting them back in the forests. And we are on the precipice of being able to restore a tree, possibly just in a matter of a couple decades, thanks to that technology. Chestnuts reproduce through pollination, so they're able to take their single line that they engineered and cross-pollinate these surviving root systems and other chestnut trees. And I got to see a presentation by the team that does this, and they're out to 2,000 different outcrossings now. So they will have a genetically diverse, restored population of this tree that only took a single gene to get back. And while that doesn't have a climate change angle, that's really the inspirational project for everyone trying to work on biotech for conservation, is the idea of, can we find similar scenarios where a single gene can help a species overcome its biggest challenge? Is there a single heat resistance gene to help certain types of corals? Could we get them from heat tolerant corals and pass them to lesser heat tolerant corals? A most recent preprint that's worth reading by Eric Iverson, University of Texas, is on the concept that we could potentially edit and change mitochondrial DNA in species and the nuclear genes that interact with mitochondria to help them overcome climate issues. We've been discovering through science that while the mitochondrial DNA is this tiny little piece of DNA, it's 1,000% of the entire genome that you've got, and it evolves differently. It's highly necessary for physiological functions, but and people have thought for a long time that's really just intracellular machinery. Turns out that those physiological functions in the cell that it's very tied to are under high selection for climate adaptation, particularly to microenvironments, elevation, oxygen levels at different elevation, temperature levels at certain microenvironments. And so there may be a lot of room to edit the mitochondria species to help them overcome climate change, particularly species that are in what's called the escalator to extinction. These are usually mountainous species, sometimes called sky island species, because they're living in high mountainous regions. And as it gets hotter, they've got nowhere to go but up. And eventually the mountain runs out, just like the birds in Hawaii. Eventually the mountain runs out. There's no safe refuge. Those are species where mitochondrial gene editing could help. So I think there's a lot of potential here, but really the science is very new. Understanding the genetics behind climate adaptations is very new. And with the urgency of climate change, that actually, I think, is a huge compelling reason that we need to get as many of these species in zoos and captive breeding programs as an insurance policy, so we can have the time to figure out how to help them and develop this science, as well as taking samples and putting them into biobanks, cryopreserving tissues and things, that we can buy ourselves the time we need to discover what are those key genes and those key things we can edit that will make those species able to get back into the wild and live in a world that's been fundamentally changed at a rate that has been just so rapid. And I hate to go on and on like this, but I say, a lot of people will think, well, why do we need to help these species evolve? They evolved through climate change before, right? And it's like, well, but we all know the climate change we're experiencing now is so much drastically faster than ever before. I don't know if you talk about it a lot on your show, but the two other things that are just devastating to wild species is not simply that the rate of climate change is higher, it's that they are getting hit by rapid climate change after hundreds of years of being decimated and us terraforming this planet to a completely different geological state. Species once moved freely from one climate optimum to another as it moved because there was nothing in their way. And today there are 4-lane highways, agricultural fields, urban centers. There's light pollution getting in the way of migrating. There's sound pollution getting in the way of breeding. And all of them, after hundreds of years of over harvesting and introduced species and disease, have been crippled to such small levels that they've lost genetic diversity. They've actually lost the building blocks of evolution, because you need several, you go back to Darwin, you need a few things to adapt and evolve, and one is variation. And when your species loses 90% of its population because it's been hit by a human activity, you've lost 90% of your variation. You've lost your ability to rapidly adapt because you're just very, inbred now. And that's going to be something where gene editing comes in to help save the day, hopefully.
What could go wrong? Are there physical, environmental effects we should be worried about? Are there economic or social effects that biotechnological interventions for conservation purposes could have?
Oh, what could go wrong? That's the common phrase of the question, right? I mean, what could go wrong? What are the unintended consequences? Anytime we're breaking new terrain and we think, oh, what is this new frontier is going to be crazy and unlock Pandora's box. And in truth, with biotechnology, the funny thing about it is it's becoming more and more precise. And the more precise a change you make, the more knowledge you have before and after. The more precise the change, the more easier it is to measure the impact after. But of course, the more precise you make the change, the more knowledge goes into developing it. And so if we find a gene for disease resistance in a species, the one thing we worry about would be whether or not once you get it in the wild, the disease evolves, right? And that's something that people will fear. Like, what if we make a super disease? Well, we already do that in human medicine. We have our vaccinations and our medications or antibiotics, and we have created super diseases. So that's why people think of this, right? Like, oh no, what if we create more super diseases by putting the selective pressure on these diseases? The beauty of biotech versions of this is we can create much stronger selection pressures against the diseases to evolve. And if we hit them hard enough and fast enough, we can eradicate the disease potentially. We can also repeat it. The moment we see the disease starting to evolve, we can go back into the species and repeat and create that new next evolution. But a lot of people are actually approaching this from the idea that if you produce multiple alleles for disease resistance and give your species a bit of diversity first, then it will actually go through the selection itself, right? And you'll start restoring a bit of balance. We often terms in our work actually worry more about the risks to the species themselves than to beyond to humans and things, because the risks to humans are actually quite low. The risks to whole ecosystems are not necessarily that high either. There's only two outcomes to trying to help a species, and that's one, well, I'll say three. There's three outcomes. One, you fail and it goes extinct. And then you have the fallout of extinction. Two, you succeed, you reach, a nice population balance and things start to play out the way nature plays them. Third, the species does so well, it overpopulates and starts creating problems in its own habitat. That's really not that much of a concern for several reasons. First is that it's very difficult to happen. We're often working with species that are at quite a rare state. Two, when planning for these things, you would be looking at the mitigation. Are the natural predators here? Do we need to bring back predators? Are there checks and balances in this ecosystem? And the third mitigation, of course, is we can control populations. This is more of a risk with, say, insects and things that breed quickly and are hard to necessarily monitor. But for larger species, take the passenger pigeon, for example. There were once billions of them. And in a matter of just a couple decades, with 1800s technology, they were virtually wiped out. We actually know precisely how to control that population. So, that's how conservation works. It goes through this process of checks and balances, planning for the future, monitoring, and creating mitigation. Not a lot of people know how much work goes into that because successful conservation is invisible behind the scenes. You don't see people going out and burning off invasive weeds. You don't see them going out and necessarily planting things. When you go into a wilderness area that you enjoy to walk through, you see wildlife here and there. You may not see the animal that has the radio collar on. You might see its babies flourishing. You see nature. You don't see all the work that went behind it, but it's been there, and it's been getting better and working. And that's why at Revive and Restore, we've kind of wanted to be developing this paradigm of, rather than asking right away, what are the unintended consequences? Let's ask, what if we get it right? What are the intended consequences of these interventions? Because all too often, when we delay acting, or we just refuse to act because we've been paralyzed by fear, species go extinct. And with the urgency of climate change in particular, we don't have the luxury of time to sit and debate. And I think the worry that people have in this unintended consequences dialogue is that if we act fast, it means we're acting too fast to think it through. And that's not the case. We can act quickly and responsibly by getting the right minds together. We have the technology now to run simulations, to do things in laboratories. We can act much more quickly and carefully than ever before. And we can act much more precisely to do things that are really, really fine-tuned to a species and an ecosystem. And and if we don't, we're going to lose tons of biodiversity. And while that is horrible to nature in many ways, it's horrible to the human condition. I might be preaching to the choir, but we are a species that spent 300,000 years living closely tied to wilderness and have only spent a couple centuries in an industrialized urban world where we're separated from it. And already we're seeing the impacts of that in mental health, quality of life, that we know that we really need to stop exploiting and changing the natural world and start finding a way to coexist and allow the natural world to flourish in our environments, because we need wildlife and nature to just sustain the quality of being human.
I want to circle back to intended consequences, which was the subject of a short essay that caught my eye a couple years ago. We'll put up a link in the show notes. And I want to read just a bit more than the opening paragraph, because as someone who's involved for quite some time in debates over the role of advanced, cutting edge, novel technology to combat climate change itself, this I guess resonated with me. And it opens: as the biodiversity crisis accelerates, the stakes are higher for threatened plants and animals. Rebuilding the health of our planet will require addressing underlying threats at many scales, including habitat loss and climate change. Conservation interventions such as habitat protection, management, restoration, et cetera, have the potential to help threatened or endangered species avert extinction. These existing, well-tested methods can be complemented and augmented by more frequent and faster adoption of new technologies, such as powerful new genetic tools. In addition, synthetic biology might offer solutions to currently intractable conservation problems. We believe that conservation needs to be bold and clear-eyed in this moment of great urgency. Finally, the substantial history of intervention should encourage us to thoughtfully pursue novel approaches to conservation as the technology advances, focusing on the future we want rather than being daunted by the future we fear. End quote. Thinking collectively about the scientific and environmental community, are we excessively daunted by the future that we fear in a way that prevents us from focusing clearly on the future that we want?
Absolutely. In fact, humans in our nature, for some reason, are so focused on the things we fear that we just create self-fulfilling prophecies. We're afraid of all these unintended consequences, and that cripples us from doing anything right in the moment it needs to be done. And then when we turn around, the thing we feared is right in front of us. And it's kind of an odd thing. I think there's a great conflict in the human mind and state that we are simultaneously explorers willing to take a risk. You think of how we got to where we're at. People literally put the value of finding a new land above their own life for that thrill of exploration. And as we settled the world in multiple waves, from the first waves of indigenous peoples out of Africa to everywhere, to then the different waves of colonialism, as we created more permanent habitats and environments through agriculture, a sort of weird provincialism began to creep into human beings, in which we no longer wanted to explore. We wanted to hollow up into our own little safe corner. And I think there's a great conflict between those in many human minds. Whereas, yeah, we would love to discover more science as long as we're safe in our home and everything's fine. We have done great leaps in astronomy and physics and other things where we've been able to hand the wheel over to robots to do the discovery for us and watch from our laptops. Biology doesn't do that for us very well. We're using drones to monitor species now and see amazing things, but biology is much different, and the conservation needs are much more demanding of involvement, of getting your hands dirty. And the problem is, you can either get your hands dirty and solve the problem, or be afraid of taking a solution and have the blood of those species on your hands afterwards. Like there's no way to come through it clean. You either get dirty physically or you tarnish yourself emotionally and psychologically, if you're a type of person that actually cares about the biodiversity of life on this planet. I don't know when that kind of fear of unintended consequences crept into the human psyche, but it's heavily there. And I think it's related to the core problem in our brains in society that makes us racist, sexist, homophobic. All these things that create fear in us of what's different is really a fear of change of the status quo of, this is how I like my life right here, and I want to keep it that way. And anytime that's alienated or threatened, our immediate response is going to be somewhat of fear. And when we can't justify the fear, then we lash out in hate and anger. For conservation's purposes, you see all of those at play. Whereas people will point to invasive species of the 1800s and say, see, you meddled with nature and it screwed up. We have concrete proof here. And they're taking those things out of context. They'll say things like, oh, well, you're going to do these interventions, but you're going to disadvantage the BIPOC community. This will just be things for more rich white people. Or you are going to create problems between those communities. We have a wide variety of things that keep getting thrown up as roadblocks, oftentimes not by the people and communities that will be affected, but by people that are just disturbed by the concept that their concept of life may change. And I would love it if we could get the human species back to a place of awe and discovery and exploration, and a species that embraces change as being something beautiful, that we're a species willing to look at metamorphosis and say, okay, I will take the risk and see what the next chapter is. Because if we don't change literally everything about how we create food and acquire energy and use land, we won't be able to meet the challenge of the biodiversity crisis, and we won't avert climate change. And people everywhere, no matter their status, will be negatively affected.
Does this leave you optimistic about the future? Do you think that this is the course that humans may take over the next century in controlling and managing, more accurately, crises such as biodiversity and climate change?
I am in a constant state of limbo personally, with my anxieties over many things in the world. But several years ago, I chose to bring 2 children into the world. And no matter my own anxieties and fears, I have to fight for optimism and that hope that the world they might inherit and my grandchildren may inherit may be better than the one we live in on so many levels. And biotechnology, at least in the realm of conservation, offers a plethora of opportunities and solutions to problems we have today, and even ones that will emerge, that at least in one sector of this thing we need for our world and our human minds, the natural biodiversity around us, we could get to a point where not only we've saved many things today, but we've changed our industries of how we do things. We've been able to restore more wilderness, live in more coexistence, and get to a place where we actually have many types of species that we've lost for a very long time, and have just a more enriched world. And I think It's going to take overcoming our hates and our fears to get there. It's going to take rebalancing the risk-benefit equation to actually stop being choked by risk and actually look at risk as in what's the probability? Can it be mitigated? How do we proceed? What's the risk of doing nothing? And what is the benefit and value of acting? And each alternate version of acting. Being able to do that and have those conversations calm, collectedly, and at the pace that's needed, and then roll those out, will require a world of people that can work together and settle and set aside divisions. I think reaching for a world that values nature is one that embraces that cultures all around the world have ties to it, that we can actually mingle our overcoming our socio-political issues with those of the biophysical world around us. And when I talk about that future I want, it reinvigorates the hope for optimism that I want to fight for. Even if it doesn't happen in my lifetime, I want a generation to be born that has a more diverse and beautiful world for a change than the ones that keep being born that has less and less in it. And that's a world that will truly be able to find harmony in many levels.
Our guest in this episode of Challenging Climate has been Ben Novak of Revive and Restore. Ben, thank you for joining us.
Thanks for having me.
Well, thanks for listening. Please rate or review us on Apple Podcasts and elsewhere, and consider supporting us on Patreon. That's patreon.com/challengingclimate.